All right, thank you so much, Kathy. Make sure everyone can hear me in the back. So my job is to keep you awake after that wonderful meal for the next 45 minutes or so. And Kathy thanked everyone in the room, but I had such a wonderful time yesterday just beginning to tour the museum. I'm here to celebrate brains and enrichment and education. What?
I think you'll have to use the mic.
Okay.
That is just recording you, so you'll have to pick that mic up and use it.
Oh, okay. That is not what I understood. So you couldn't hear me. Now, can you hear me better? Okay, all right. So as I was saying, I'm here to celebrate brains and education and enriched environments, and the museum is doing that. Found myself feeling like a student yesterday, and it was wonderful, and I think that we owe Kathy a big applause for everything she's done to lead this program. She's thanked everyone in the house, but she's doing a great job, and thank you to the team. So I'm thrilled to be here and talk with people who are interested in the history of psychology and psychology education, and maybe a little bit about brains, reason you're here. And it's also an honor to do anything associated with Ben. And when I can't even remember the year, I think I was a graduate student when I attended this little conference, regional conference, the Southern Society for Philosophy and Psychology. He gave a talk about Harry and Lita Hollingworth, and it was even outside of my area. It was applied psychology, the Coca-Cola trial. Well, it was kind of in because it was caffeine and what it did to the brain and mental output, and I was just at the edge of my seat to see someone who could bring science to a story and stories to science. So I've been chasing a good story ever since then and wanting to read everything that he has written and used his book. My favorite was the Letters book, and when I taught history of psychology. So thank you for your contributions. A lot of personal contributions. So these are my colleagues. That might be an upgrade for some of you or not. So the rat, and I've been working with them since graduate school. I will share my journey. I'm expanding beyond them a little bit, but they have been great colleagues, always smart, always outsmart me. And the theme, as Kathy was saying, the theme of my lab, if I could capture it in a phrase, would be experience-based neuroplasticity. And neuroplasticity is how our brains change. If we're living, our brains are changing. As I tell my students, from the womb to the tomb, our brains are changing. And as we learn about how that happens, it makes us more accountable, I think, for what we're doing with our brains. So maybe it's okay to spend some time scrolling through and looking, but we need to understand more, and I think there's so much more we can understand. And I love working with behavior as a behavioral neuroscientist. That's one of my lenses, but also the beauty of the brain. I know my slides may not be very visible to those in the back, but just to be able to see those cells, those neurons, and think about how it generates a personality, a behavior, that will keep me going for the rest of my career. So this idea that our brains can change, there have been different ideas about that. And I want to approach it through three kind of different lenses because I'm here, the center of the history of psychology. I love to bring in a little bit of history and then experience or habitat, how that influences our brain, and then hopefully end a little bit on a positive note talking about hope and how the rats are teaching me a thing or two about hope. So before we knew anything about the real infrastructure of the brain, there were some thoughts about whether or not it was malleable. In the museum yesterday, I saw the exhibit of one of my favorite characters in the history of thinking about brains and psychology and such, and that's Franz Joseph Gall, who is associated with phrenology, this idea that bumps on your skull can give us some information about what your abilities are. We don't really have bumps on our skull, but he was going for something interesting. Well, his sidekick, Jan Spurzheim, promoted him, but he had some ideas about plasticity. He said, it may be asked whether the organs increase by exercise. This may happen in the brain as well as in the muscles. This idea, use it or lose it, already in 1815. And as we learned what the structure of the brain was like, what those cells looked like, it was just mush. The perspective was just this 3.5 pound of mush. But the Italian neuroscientist Camilla Golgi discovered this stain that stained the infrastructure so we could start to see the neurons. And then Santiago Ramón y Cajal in Spain, who was an artist and a physician researcher, good liberal arts. He spent a lot of time looking through his microscope. He supposedly took it everywhere he went, total nerd, out of town or whatever, and sketched and sketched and sketched. Together they won, they were recognized and won the Nobel Prize in 1906. And I grew up in Alabama, so I would say, bless his heart, because Golgi, even though he discovered the stain that allowed us to start to see that the neurons were separate from one another, he still had this idea of it being this continuous neural net. And so he gave his talk talking about the neural net, which was so wrong, but he discovered the stain. But Cajal kind of won the day, looking retrospectively. Even though he couldn't see those tiny gaps because he did not have the microscopy power at the time, he had the insight that there were separate little units. And so he advanced the neuronal doctrine, which was true, that they were individual units. And this is a picture of his drawings. I stain brain cells in my lab, and they look just like that. His work is used, it's contemporary today as it was then. And once he started to see this structure, and he's been a little all over the loosey-goosey about the plasticity, but he did say any man could, if he were so inclined, be the sculptor of his own brain. We would extend that to everyone, of course, but I love that idea of being a sculptor of your own brain, and this was in his advice to a young investigator in 1897. So from the neuroanatomical perspective, we were starting to think, well, maybe the brain can change, and certainly we know that it changes until development, and when development ends, childhood or whatever. The idea was wear your helmet, don't do drugs, don't do whatever can kill neurons, because those are the only neurons you're going to have. That's still pretty good advice, but can we change our brains? Well, Darwin, looking at the naturalists, he noted that the rabbits that they were selling at the market that were selectively bred, their skulls were a little smaller than the skulls of the wild rabbits. And that could be a selective breeding thing, but it could be in response to their lifestyle, because he said, I think it's hard and dangerous and complex being a wild rat. So maybe their brains were bigger and their skulls were bigger, to think that how you engaged your brain may influence the brain. I'm a huge fan of Beatrix Potter, a wonderful children's author, but also a great naturalist and conservationist. And she noted that the fancy mice that were bred as pets weren't quite as smart as the wild mice. And I'm not sure if she's holding a fancy mouse or a wild mouse. So she was noticing that maybe the more complex the environment, that may change the brain and the output and mental abilities. It started to hit the field, getting a little bit closer to psychology in the 1940s, and we've got some Canadians here, with Don Hebb, known for writing The Organization of Behavior, such an important contribution to the field. But he started writing about kind of anecdotal evidence in some of his papers and talks about the home-raised rats, that he took some rats home to his daughters, and he claimed that those rats were smarter than his lab-raised rats, suggesting that the environment, and there was more going on at home, led to increased learning, which he would have suggested would be some changes in the neuronal networks. And then my hero, Marion Diamond, working at UC Berkeley in the 1960s, working with a team. There were 3 others. She was the neuroanatomist, but there was a neurochemist and neurophysiologist. They had read what Hebb had written, and this influenced their thinking, and so they wanted to put it to the test. And this is in every intro to psych textbook, but they conducted these studies with what they call an enriched or complex environment, so bigger cage, more animals, more things to do, and they would also introduce new objects, so there was some novelty there. And within a month or two, these are youngish rats, they were post-weaning, she noted that the cortex on the outside of the brain was a little thicker, statistically significantly so, in the enriched rats compared to what they called the standard lab rats. And I was able to interview her before she passed away, and she told me that when she first presented those data in the later 1960s, and you can imagine she was probably one of the few women in the room, so she was at the podium presenting these data she was so excited about. And she said, we'll call him a gentleman, stood up in the back of the lecture room and said, "Young lady, that brain cannot change." Well, she was a good scientist, and she said, but I'm, like any good scientist, I've replicated the study, and it does seem to be a robust finding. So it appears that this audience member, maybe his brain was not changing fast enough to keep up with what was going on. And since that time, because it really was such a interesting finding, there have been thousands of studies looking at enriched environments, changing the stimuli, the animals. It's messy research, so some people push back, and they still do. How do you know exactly what is changing the brain? But life is messy, right? It's hard to just identify that one variable all the time. So it's somewhat of an imprecise sculptor, but a sculptor nonetheless. So there have been a lot of different dependent measures, but this is just one looking at that beautiful neuron. This would be an animal in the complex environment. And so you could count the number of processes, the arborization, or the length of these processes. My students can spend hours and hours and hours tracing and counting. Am I still? Okay. So there's been so much found with just putting an animal in an environment and looking at how that changes the brain. But if the environment is so important, does it matter if that environment's in the lab? And I started to think about, what is this Umwelt, this ecological environment that is appropriate for species? That's the German word that was used. Is the laboratory Umwelt too different from the natural Umwelt? And this is a question that is worth thinking about. So we tried to, I was able to ask Marion Diamond, because my students, I learned so much from my students, and it's so great to see students here, and keep asking these questions to your professors, because we get in a rut sometimes about the way we think. But my students said, well, why do we always expose the animals to these plastic kind of artificial toys? Like something you get out of a Happy Meal in those days, little mirrors and metal things. That's a great question. I've never really thought about it. It was convenient, and we did it, and we washed them. And I asked Dr. Diamond, and I had read, it was the cover story, I found it in the 1972 the Scientific American edition, and she talked about putting some enriched rats outside at UC Berkeley in a sand pit or something. I said, what did you do, and do you think it's worthwhile trying to look at more naturally enriched environments? And she said, yes, if it's a small sample, their brains look larger, do it. And when Dr. Diamond tells you to do something, you have to do it, right? So we started to compare our artificial environments here with all the usual artificial stuff, rubber balls and such, matching the number of items and the function of items in a natural habitat. So we had kind of dirt, they love to dig, and if there was a plastic ladder, we would have a little twig ladder. And again, this is the standard government regulated way to house rats. I don't know what it's standard for other than a lab. They can't even rear up. There would be food and water. So this has been troubling me for a long time. So we kind of called these the city rats and the country rats to keep Beatrix Potter. And we've done lots of studies. We found when we give them like special operations soldiers tasks where they have to figure out how to dive down and they remember there's a gap here to get to that Fruit Loop, and that's the currency of my lab is Fruit Loops. I should get them to fund my research. But when we look at an area of the brain that's involved in reward and pleasure and motivation, the natural enriched rats, when they're faced with this challenge, it seems like their brains were saying, I've got this, I can do it. and maybe this makes them persist. We're also very interested in stress. I don't know if you've ever felt stress. It's very much a part of us, and we have this weird relationship. We need to have stress to survive, but not maybe chronic so much. So you have probably heard of cortisol, the adrenal steroid. So we have some areas in the brain that tell us that this is a threat, and it sends a signal to our adrenal gland, and then it secretes this cortisol, which is good. It helps distribute energy throughout our body so that we can respond to that threat and survive. I think too often we find a single biomarker. We make things a little too simple. There's another adrenal steroid, DHEA. It's a long name here. You can actually buy it in a health food store. I'm not advocating that. But your body makes it, and there's even some human research going back to special operations soldiers, and when you go through intensive training, that you have higher DHEA to court ratios. And it appears that DHEA may mitigate or provide a buffer against the toxicity of those stress hormones. So we wanted to look at that ratio in our country rats and city rats, and you see that we're seeing or have seen a higher DHEA to court ratio. So if you're going to have the stress hormones, that you have that buffer as well. And you may think, well, how do you measure stress hormones in a rat? A lot of people will test it in the blood, but it's very invasive to try to get blood, and I don't like to do that. So they're very happy to poop for us. So it's quite glamorous to work in my lab and to process rat poop. It's a delay, about a 12-hour delay from experiencing the stress to when those metabolites end up in your poop, but you can learn a lot about physiological measures and poop. And I've also worked with some beach mice, Peromyscus polionotus, and where I grew up, part of my childhood around Gulf Shores in Alabama. This is sometimes called the Alabama beach mouse, and it's white and it just blends in with the sand. We had these in my lab, and a very brilliant vet tech, Carol Stevens, it's just funny what I don't think of as a scientist. She said, "Kelly, here's an idea. Why don't you put sand in the beach mice cage?" Duh. And no one's done it. So we did. And it was like, okay, we're going to turn the on switch on for these animals. They've been on pause the whole time they've been in the lab. And it was as if they had hired heavy excavation equipment to move everything around. I couldn't even recognize the cage. They were so busy, so engaged. It is important to have the proper environment for these animals. And it makes me think. I was reading, I think it was an Atlantic article about Jack Fisk. Now, he lives in Charlottesville. I don't know if you've heard of him. He's married to Sissy Spacek, the actress, and he is a design producer for films. A recent one was Killers of the Flower Moon, with Leonardo DiCaprio, about the Osage Nation and how these individuals were manipulated. But he wrote something fascinating. He said that he spent so much time studying the period, the time period for the film. And even though you could only see the table and what's behind it, he would meticulously look at what should be in the drawer. So if the actor opens the drawer, he wanted the exact correct little items. And the way the dust was on the road back then in Oklahoma, and the little screws in the furniture. And his idea is that more relevant and exact and accurate that environment is for that time period, the more authentic the performance would be. And it made me think, and when we were looking at this, you may not be able to see this very well, but I was able to work with a wonderful illustrator for my Wild Brains book for some quirky images. As a scientist, we're the set designers for our animals. It's our responsibility not to just put them in a plastic cage and watch them and say, action. And so I've started to think of myself as more of a set designer. And another researcher talked about how important it is to measure what animals are doing on their own accord, not what the only one behavior that we're forcing it to do. Some of that may be helpful, and I've been doing that my whole career, so I'm pointing the finger at myself. And it's also interesting to think, and this is a little bit different from my lab, animal lab, when those landscapes are no longer there and the impact they have on what's inside our brain when the landscapes become mindscapes. And this is a different area, but when I was writing Wild Brains, I was interested in how animals act in captivity in the last chapter. So I was reading about that and how their behavior may change and stress. And then I started, humans are animals too, how this impacts humans. And I had seen, we get data and stories everywhere. I like to watch this Dateline every once in a while, mysteries, and talk about behavior. But there was this, they did 2 episodes on Eduardo Valseca in San Miguel, Mexico. And he and his wife, he had a ranch. His father was a well-known publisher in Mexico, but he was no longer around, and Eduardo really didn't have a lot of money, but some people thought he did. He and his wife were driving their 3 small children to school one morning, and after they dropped them off, they were kidnapped. They let Jane go after a while, but they kept Eduardo, and I know humans have been through horrible situations, so this is just one, but I could not imagine what this man went through. They kept him for 7.5 months in a box barely bigger than his body. He was nude. He was starved to half his body weight. Bright lights were above him. He was recorded, the camera, so he was never on his own, and loud music played for 19, 20 hours every day. And I wanted to know, what does a human brain do when the landscapes have all been removed and the fear? And they also shot him twice because they were trying to elevate the concern. They would send pictures of him. He was shot in the leg and then the arm to try to get the money, but the family didn't have the money. And this is him toward the end. You can see the big difference. So I tried to be an investigative reporter. I said, I want to talk to this man. I want to know what he thought, what was going on in his brain. So I kind of figured out what his kids' names were from the episode and went through social media and said, I know this sounds like I'm stalking your father, but I'd love to talk to him about his experience. About 6 months later, I got a call from Eduardo. We had the most amazing conversation, and I asked him, how did you survive minute to minute every day for 7.5 months? And he said that he repeated 3 mantras over and over and over again, thousands of times a day. One starting with thinking of the universe and that there was some organization to the chaos and nature, and his ranch, he could see the stars, so he would envision that. And then his family had 3 small children, and Jane, thinking that love could conquer all. And then he would think about, they've done everything they could to my body, but I'm the boss of my brain and my mind, and just kept reminding himself of this. He also thought of something that was his most cherished memory. Jane would make banana pancakes on Saturday, and so he would dream about the banana pancakes. And in his dreams, because his body was starving, he said he would dream about opening a refrigerator and just be full of food. So even at night, he could see and maybe experience something like eating. He would hallucinate about protein, almonds being slid into the box, and that the mindscapes were kind of keeping him going. I invited him to campus just last fall, and we had a fireside chat talking about resilience. This man is the most optimistic person I have ever met, and the students just loved hearing his story and how mentally, healthy he is. Not that everyone should be, but it's just interesting when everything's taken away. Our experiences up to that point can influence our experiences going further. Okay, so I was kind of feeling good about myself with my naturally enriched environments. I was going above and beyond in the enriched environments. Good for me. Until a wild rat literally broke into my lab. You can't even make this stuff up. And I had to report it because you're not supposed to have wild rats on campus. They carry things. And so they hired a professional. And after 3 weeks of them not being able to catch this one rodent in my lab, I said, this animal is so different from the animals I have been studying my entire career. I need to start going to the wild. I need to go wild. So I called the pest control people. And I said, where are people complaining about rats? Because, ma'am, you want to go to the rats? Yeah, I want to try to trap some rats. I think I can do this. So my husband would drop me off around the airport. I had my snake light on and traps. And it was as if the rats said, thank you for the apple and peanut butter, but we're not stupid enough to go into your trap. So they truly were smarter than me. I had such respect for these wild animals. And then when I got to the University of Richmond, I met someone who was smart enough to trap wild rats. This is my colleague in biology, Jonathan Richardson. So going behind certain restaurants in Richmond, leaving traps, you can catch these wild rats. So they're the same species, Rattus norvegicus. They look a little different than my black and white animals, but the same. And this one is anesthetized because if it wasn't, it would be biting my nose off, which I kind of like because it's that raw, natural behavior. They have not been selectively bred to be docile, so they're easy for scientists to handle. So we started to compare. The university did not want us doing work with wild rats, but we have a little hut in the woods, and Jonathan would trap them, and not to bore you with too much data, but I was pretty amazed that hardly anyone had compared wild rats to lab rats, and so much of our biomedical research is based on lab rats and mice, more mice now. And so I just wanted to see if, does it matter if it's wild or in the lab? So controlling for body weight, we also had some farm animals donated by the United States Department of Agriculture. You see the lab rats had a smaller brain, significantly smaller. And the cerebellum, where most of the neurons are in our brains and the rat's brain, involved in movement that's so important for brains, they had significantly smaller cerebellum. And we also looked at cell counts too, and it matched these findings. But I was really interested in the stress hormones because I knew it's stressful being a wild rat. And so we started to collect some poop. This is working with Molly Kent, who is my postdoc and is now a professor at Virginia Military Institute. And so we tried to measure the hormone. Now we were comparing to, we call it relocation stress. You trap them, you bring them here, you wait, have this 12-hour delay. But we also order our lab rats from this lab rat factory. They're put on a truck, and that's kind of stressful too. We don't think about it too much, but and is the microphone okay? Is it echoey, or it's okay? Is there anything I can do to make it better? Okay, nothing. It's okay. Okay. Let me know if it's too funky. So we compared the lab rats and wild rats with to look at corticosteroid, their version of cortisol. Now, their stress hormones were so high that the kits that you measure for assessing stress hormones in rats in the lab, it wouldn't work for a wild rat. So we had to work with the technicians to dilute our samples. That didn't make me feel too good that we were studying something real with our lab rats. And the females here in the blue, we could not control for reproductive history because they can't fill out forms in the wild. So, but we've replicated this about 4 or 5 times, and we generally find that the wild rats' stress hormone levels are about 6 times higher than the lab rats. And if I was looking at a lab rat with stress hormones levels that high, I would say you should be dead because this can't be good. But these wild rats are ripped and healthy and big brains. I don't know how they're handling it. Maybe this is a secret that they can tell us. And one of the reasons I'm interested in wild brains. They're also the adrenal glands. And I have to say that Kurt Richter at Johns Hopkins around 70 or so years ago, he said that he trapped 10,000 wild rats, so that's a lot. I don't know where that's logged, but 10,000 wild rats, and he measured the adrenal gland, and the heavier the adrenal gland is, that's a sign or a clue that it's pumping out more stress hormones. And he reported that the adrenal glands were about, this is counterintuitive here, 3.5 times larger than his lab rat adrenal glands. And that's about exactly what we found 70 years later. So they seem to be very stressed. So I have not shared all the really interesting differences between the wild and the lab animals of the same species, but there are a lot. And I don't use albinos, but one of the most popular models is the albino, which is even less representative of a wild rat. So if we can't generalize from a lab rat to a wild rat, I'm not sure generalizing from a lab rat to a human makes a lot of sense. And again, I've been doing this my whole career, so I'm pointing the finger to myself. This made me think, how in the world did we land on rats? And again, it's mice now. How did this start? So this took me down this really interesting rabbit hole. Meet Jack Black. He was the royal rat catcher about the mid-19th century in London, where rats were a problem. They were spreading diseases. People didn't like them. So they would pay people. You get paid a certain amount for the number of rats you could find and bring in and kill. So he was very skilled, and he wore this green velvet jacket and this little sash, and very proud of what he did. And he had terriers that would dig in and find the rats, and he just had this whole system. And then he also, in London, there was this education. I think the fact that I have two microphones on might be, but there is this, for entertainment, you could go to a pub and they would have terriers. This sounds horrible, it's not consistent with animal rights, but I'm just reporting history here. So they would take bets on which terriers could kill the most rats when they were out drinking and in the pubs. So Jack Black was good at catching rats, and he thought, if I started breeding these rats, I can sell them to the pubs. I'm getting money for the ones that I'm killing. I'm selling some, I'm breeding them on the side and selling them. And then every once in a while, a white rat would pop up, the albino, and he said, I think the elite ladies would like a little white, majestic-looking rat. So he sold those for the pet market, which may have been, ended up with Beatrix Potter and some of her early fancy rodents. And so he'd carry them around in a little bird cage. So he had a system here. Okay, so that was going on then. And then in psychology and psychiatry, there was some talk about we need some models, good question, to test and assess some of our ideas about what's influencing behavior. Swiss psychiatrist Adolf Meyer, who came to the United States, well-known psychiatrist, he was thinking about models. Henry Donaldson was a director of the Worcester Institute, and they wanted to institute a model. And to their credit, they were both interested in comparative and differences among different animal brains. But like me, they were not good at trapping wild animals, and they had kind of thought a rat may be representative because they were available. And then Meyer said, well, they're breeding these rats in London. So he brought about 4 albino rats over to Hopkins, and they became a significant part of the stock, the genetic stock, this albino animal for breeding. And they referred to these as the Worcester rat. And then we just had this industrial complex. NIH, they were funding research, but you needed a standard animal model. They thought that the white rat looked standard because it wasn't any variation. So they weren't interested in individual differences at all. Which is, if you know anything about research, we usually respect individual differences and representation. Joe Garner at Stanford wrote this about how we have used rats in our research. We would never perform a human drug trial in 42-year-old white males with identical educational levels, identical socioeconomic statuses, identical jobs, identical houses, with identical and locked thermostats, identical wives, identical diets, identical exercise regimes, in the same small town in Wisconsin, who all incidentally had the same grandfather. So can we realistically expect mice, and rats too, in exactly this kind of Stepford experiment to tell us anything about humans in general, our variability in risk or response in particular? Ouch. Yeah, it's interesting to think about that. Now, human researchers, I'm a rat researcher, not a human researcher, a human doing rat research, they've been called out about using weird, mostly Western, educated, industrialized, rich, democratic populations that represent maybe 12 or 80% of the research participants, may be representing 12% of all human population. So there's been some effort to change that, but not so much. We like our animals to be exactly the same, so that one variable we changed, we feel confident that that had an impact. So I'm going to be the first to line up in that weird kind of queue and call myself a recovering laboratory animal model exclusive researcher. I'm a lame researcher. So exploring, learning about how just different habitats can change the brain, it makes me also think that there are different animals out there that we could probably learn from as well. And this is what I'm calling the Neuroverse. I've already talked to you about the wild rats, which is a different animal than the lab rat. But I had the distinct privilege of meeting and working with Dr. Patricia Wright, biological anthropologist who found the funds to create the Ramaphana National Park in Madagascar. Now, Madagascar has some really interesting animals. If you've seen Disney film Madagascar, you know about the lemurs. And I don't know if you can see, but this little mouse lemur that was a character, they've become kind of a hot research model for Alzheimer's disease. It's interesting also, pointing the finger at myself, I don't necessarily do Alzheimer's research, but most of the Alzheimer's research is done on mice, perhaps the shortest lived mammal on the planet, 3 months probably out in the wild. These guys are more like primates, kind of pre-primates the lemurs are, and they probably live 8 to 13-ish kind of years, so they may be an interesting model. So I was able to go to Madagascar to start to look at how can we measure cognitive abilities. I won't go into the details, but we kind of they live trap them and go into the doctor to get their medical parameters, stress hormones and such. And then we keep them kind of like in a lemur Airbnb for a couple of days and ask them to do. So I came up with no tools at all, but some associative cues related to bananas and created a foraging tree. And boy, you can tell they're the size of a mouse, but they are acting like a primate. They are all over it. They do so well. They just look like little forest fairies to me. It was just magical to be there. And we hope that we can get some more funding, not to bring them into the lab, but to see what we can take the lab to the wild, to nature, to see what we can learn about them. And they were already teaching me some important lessons. And then one of my favorite mammals is the raccoon, even as a child. Their curiosity, their forepaws that are representative of their motor cortex, similar to our hands. Now, before the lab rat was the lab animal, there was some research in Oklahoma around 1900. Lawrence Cole started training raccoons. He thought they would be an excellent model to learn about learning. Now, it looks like he's kind of teaching them circus tricks here, but there is some research. And I think one of my favorite descriptions in the research methods section of a research article was related to this. They said all of the research participants escaped through the ductwork. And I can imagine these raccoons with their headlamps on and the plan spread out. So for the the essence of what we wanted to study, curiosity, cognition, problem solving, they were too good at it. It was easier to do the research with the rats that seemed perfectly happy to sit in those cages and would reproduce. So we abandoned the raccoons. But can we do some research with raccoons? I was able to partner with Dr. Sarah Benson Amram, who was at University of Wyoming at the time and is now at University of British Columbia. She had done some hyena work in Africa, and the raccoon became her species when she came back to the U.S. So she was working with the U.S. Department of Agriculture, who were looking at testing some rabies vaccinations and able to do some behavioral work with the animals so we could look at problem solving, look at flexibility, and we had some that they would teach them, you turn the knob to open and then you would lock it and then they had to switch and be flexible. And out of about 21 animals, about a third of them could solve it all, a third were kind of intermediate, and a third didn't seem interested or didn't engage so much. And so that was interesting. We had some groups, and even though I could not find a proper brain atlas, we call that pictures of the brain's rat atlas or a mouse atlas, and it shows us all the brain areas for that species. I don't have that for a raccoon, so I'm kind of working blindly here. But look at that brain. Now, I don't know if you think brains are beautiful, especially after dinner, but this is a gorgeous brain. Look at those squiggles, that gyrification. So a rat brain is very tiny and this has a smooth cortex. They still outsmart me, but with this gyrification, their brains were already looking very impressive. And we did find by looking, and again, beautiful brain. This is the hippocampus, the dorsal part and the ventral part. the solvers had more neurons in the hippocampus that's involved in learning and memory. And I've done some collaborative work with Susanna Herculana Huzel, who was initially in Brazil and now at Vanderbilt. She has a fancy way of counting cells. If you ever are bored, you want to Google something nerdy, do Brain Soup and TED Talk, and you'll hear an amazing talk. So I collaborated with her to be able to do some cell counts to look at how many neurons they have, and she's looked at about 200 different species. So a rat brain, there's a big rodent called a capybara, about the size of a pig, but as that brain gets larger, the cells get larger, so they don't have a whole lot. Bless their hearts, right? But some primates, as they get larger, as we got larger, the cells are tiny. They remain tiny, so we're able to pack in more neurons. And so the raccoon so far is the only non-primate mammal that's able to do that, to pack in more neurons and scales like a primate brain. They're not a primate, but this is the reason they're getting into the garbage cans. We also identified this elongated neuron called the von Economo neuron. This has been found, you can see it, this very beautiful neuron here. Maybe a fast projecting neuron. They found them in apes, and larger, we first thought, oh, we're special, the apes have them, but then they found them in some large animals. But we were the first to show them in a raccoon, and I'm wearing a von Economo necklace right now. An artist made that for me. So we're not too sure what that means, but it's interesting. Again, some Canadians, I've become good friends with Dr. Haley Heslin at the University of Saskatchewan. Her day job is being chair of the Department of Agriculture and Resources, and her other job is she has a research bandit where she takes care of abandoned raccoons, up to 60 a season herself, bottle feeding. And I got to just sit in the middle of them for a week and just give them like preschool, what would you do with a slinky? The whole put cotton candy in a pool and it disappears and they're looking for it. But it's just such a whole new area for research. We don't know anything about raccoons and their brains. We don't even have the tools to look at it. Again, I'm not advocating bringing them to the lab, but how can we bring the lab to them? And one day I want to write children's books because I have to write The Raccoon from Saskatoon, which is where I was there. And being in, you may have seen this on the news, but just a few blocks from where I started doing this raccoon research at Randolph-Macon College, the ABC store in Ashland, Virginia, reported they went in, so the workers went in, this was on BBC News, so they went into the liquor store and saw this. Now, your average wild animal, so it climbed in through the ceiling, but if it knocked over a bottle, it would go, that's a scary sound, shattered glass, it would run and hide. This animal knocked over all of those and then proceeded to taste every one. And then they found it in the bathroom, passed out. So the animal control people sobered it up and then released the raccoon. I wrote about this in the conversation. You just can't make this stuff up. So my artist friend did kind of the Mission Impossible, and they had to create a garbage bin like this in Toronto. I was giving a talk in Toronto, and they were kind of clapping. They have this love-hate relationship, so they paid $31 million for a contract to design a garbage can that raccoons could not get into. For the most part, it kept them out, but a few made it in. So I'm kind of, instead, we hear about going from the bench to bedside, but I'd like to explore going from the bush to the bench to the bedside and what we can learn from animals that aren't in the laboratory, that aren't selectively bred. What are their secrets? And outside of my lab, there have been some really interesting findings. There's a little shrew about the weight of 5 playing cards that can't afford the energy to hibernate or to travel, to hibernate or migrate. And so it has to live. It digests parts of its own brain, like up to 20%. And then in the spring, when resources are available, it grows it back. What can we learn about recovery of brain trauma, brain loss, neural function from this little animal? And then many of you probably know the prairie vole story. When they were trapping them in the Midwest, the researchers thought, why do we keep seeing them in pairs? And we've learned that they're monogamous, they pair bond. And then you could take that back to the lab knowing what their natural mating structure was and understand that this neurochemical oxytocin was really, the bonding, the love, cuddle chemical that was keeping them together. If we didn't watch them and observe them in their natural habitat, we never would have caught on to this. And then some researchers were looking at the Gila monster, this reptile, and thinking, how does it go so long without eating? Let's start to explore its saliva. And they found the template for good or bad, has been recently described as revolutionary as penicillin, and that's the GLP-1 agonist that came from this reptile. So no telling what we can find by looking at wild brains. And this maybe you've heard of the lamppost parable or analogy, but if you haven't, this idea of a police officer seeing someone on his hands and knees looking for something under the lamppost. And he said, "Sir, what are you doing?" He said, "I lost my keys. I'm looking for them." And he said, "Did you lose them right here? Where did you lose them?" He said, he pointed across the parking lot. He said, "Well, why are you looking here?" And he said, "Because this is where the light is." And I think we've been looking at the rats and the mice because that's this industrial complex. This is where the light is. This is the only way I've been able to do research. I could not get tenure or have publications if I was trying to do raccoon research. But if we could only look up, we could see so many fascinating animals, behaviors, and brains. And so I hope that we can start to look up. Okay, switching to something a little bit more hopeful and quickly. Some of this neuroplasticity, this idea of changing, let's go fast, changing our brains, maybe they could be used for therapeutics in addition to or in place of drugs and surgeries. I've talked about this circuit that I think is important. When you look at the real estate of the brain, all of these areas are involved in movement. The cerebellum with 78% of all of our neurons hanging off the back of our brain. The striatum in the middle of the brain that's involved in coordinating, smoothing out our movement, and Parkinson's disease affects this brain area. Our motor cortex, and there are connections between the nucleus accumbens involved in reward and this striatum. And so we've done some studies trying to connect effort with rewards, having our animals have to work a little bit, exert some effort for those Fruit Loops they love, versus animals that don't have to. They're given the same number of Fruit Loops. So we have our worker rats and our trust fund rats that we work with. And we call this effort-based rewards. And we've done a lot of studies and published them. But generally what we found, and we're talking at lunch about learned helplessness, we found learned persistence. We give them an unsolvable problem. The worker rats will persist longer. They have increased flexibility, which I think is a huge characteristic of intelligence. Markers of neuroplasticity, the production of new neurons called neurogenesis. And I marked this area, the lateral habenula, that's involved in like a freeze response. And what we're trying to do is build resilience against something like depression. And if you're kind of stopped in your tracks, it has some face validity for depression. And also the drugs that everyone's so excited about, the psychedelics like ketamine. If you could look at what that's doing in the lab animals, they're hitting the habenula and making it less responsive. Well, the effort-based rewards are doing that as well, related to neuroplasticity. So maybe we can do this behaviorally. So I've introduced the term behaviorceuticals that I've described in Lifting Depression and Well-Grounded. This idea that we can intentionally change our neurochemistry, our neurophysiology, our behavioral output through behavior. And that effort-based rewards is kind of like a rat model for cognitive behavioral therapy, which is so effective, but hard to have models for this. And it's not research, but it's interesting and comforting, satisfying to get stories, anecdotes, like this woman in the non-scientific journal Woman's World that you would see as you're checking out the grocery store. But she had lost her daughter, and she reported in this article, reading Well-Grounded, and quilting, and how quilting made her feel more like herself as she was going through this. Or Patrick Wells, who read Lifting Depression. He was a war correspondent, and he said that he had he wasn't diagnosed by a clinician, but he had the symptoms of post-traumatic stress disorder. And he didn't want to start the and I'm not a clinician, so I'm not giving you any advice about medication or such, but he decided to build a boat with his own hands. And it just was transformative for him as he was thinking about and planning and getting the lumber and making it. And he was so impressed with how that transformed him. He started his own business in London called My Own Two Hands, where he uses his journalist abilities to make videos to teach people how to weave baskets and such, and he sends the materials. So you get the materials and the instructional video for carving and baskets and such. So I loved hearing that story. So we need more research on this and to be more translational. And then finally, as we're going through the pandemic, all the students were gone, and there's just my trainees and myself. And we had these rats we had trained to drive cars. It's been a science outreach and some interest in that. So we would take turns coming in to feed those rats, just a few of them. They're kind of like the celebrities of our lab. And we were able to publish some of this work, showing that animals in enriched environments, I wanted to get a proper publication out of it before we took it out on the road, per se. And the enriched animals learned the task. It's hard to teach a rat to drive a car, but it's kind of scary, and we did all this, not human factors, but rat factors, how to do the dashboard and all this. So they learned faster than the standard rats. But we had them in the lab. Oh, and I should say that even though it seemed a little bit light science, this was in science, and they talked about all the impressive things that rats can do. And they have a picture of our car saying that rats, based on this research, they have intentional agency. So there you go, that a rat can do that. But when it was my turn to go into the lab, the rats met me like this. They were jumping up and down and reaching out like they were excited to see me. And I had a low mood through the pandemic, and I thought, I spent my whole career studying chronic stress and depression and aggression. Whatever I was seeing, I wanted to study that. What was it? Excitement? Went to the literature. There are some studies where it may be okay to use words like joy with non-human animals, a brief burst of behavior associated with a stimulus. And like when I asked my dog, Brody, do you want to go for a walk? And he's going around in circles and jumping up and down. Then if I could see joy in my dog, maybe I could see joy in rats. And I won't go into all the details, but we designed a study to maybe bring joy and something like hope to the rats. I landed on this idea of anticipation. If you, there's the book, you give a mouse a cookie, but if you give a rat cocaine, it will, where you deliver it, it increases dopamine, that reward kind of, I'm oversimplifying, neurochemical. But what's fascinating is if you have fancy equipment to measure dopamine, it's not just when cocaine hits the brain that you see the rise in dopamine. It's when the rat approaches the bar to press for the cocaine, which suggests that the brain is not differentiating the anticipation of the cocaine from the cocaine. So I could play more with this anticipation period. So we did a lot of things where the rats had to wait for things, like give them a Lego block, wait 15 minutes to get your Fruit Loop. Get in a transfer cage, and then we take them to Rat Park. But you have to wait like you're in a minivan at Disney for 3 minutes before you go, and then get them sunflower seeds that they had to shell. And the control animals, they got all that stuff at once. They got shelled sunflower seeds. So they got the good stuff, but they didn't have to wait for it. And so far, we've seen differences in an optimism scale. You can measure optimism in a rat, where they're shifting, especially the males are shifting. Females are staying a little bit more reality-based. I don't know if that generalizes to humans or not. But increased exploration in both males and females as if they anticipate finding a reward. We can measure their ultrasonic vocalizations, and they're in the happy, chirpy laughter range. And increased brain growth factors, that's neuroplasticity. And students, they came in one day and they said, Dr. Lambert, why are those rats' tails sticking straight up? I thought, I've been working with rats for nearly 4 decades. I don't think I've ever seen this. So I put it out on social media. Some colleagues said, oh yeah, there were some reports if you inject rats with opioids, their tails would go up. It's called the Straub effect. So maybe we're influencing some of these neurochemicals, behaviorseuticals, opiates, and probably dopamine and some other things as well. So we're continuing to work. We just have one publication, but hope, you know, it's always been seen as just fluff. Maybe that's hope isn't critical for our mental health, but the brain's all in for hope. You know, the prefrontal cortex, the reward areas, movement, insula that's monitoring and our internal and external worlds, the amygdala that's approach avoidance. I'm beginning to think that it's incredibly important, and this is just a very touching study. This was a study done with Israeli children, and very sad that it's with the Make-A-Wish Foundation, and so these children had a terminal illness. And they monitored them from the time they got their wish was, oh, this is going to happen, and they were just doing their regular checkups for their mental health and their physical health, and they saw improvements. They still had their illness, but improvements in both mental and physical health after that wish was confirmed for the next 5 months before it happened. And they didn't see that in the children who were still in the queue to get their wish. So I'm beginning to think that this may have something to look forward to have hope, could be just as critical for mental health as avoiding the trauma and the stress. So it's probably, let me end with, when I was writing up this, and I called I should have said where the anticipation studies, I call it unpredictable positive event responses, uppers. And when I was writing that up, I keep talking about anticipation of positive events. Anticipation, it was just clunky, and I kept typing in. And I asked my postdoc, Kitty Hardvickson, I said, I wish we had a word for positive anticipation. We have a word for negative anticipation, dread. I dread doing this, I dread doing that, but I didn't know that we had a word. And she'd done a fellowship in Germany. She said, oh, in German they do. It's Vorfreude, and it's joyful anticipation. I think we need a little, our own word, I don't know what it could be, to make that just as important as dread. And finally, a lot of people ask me, Kelly, do the rats like driving? And so that's an interesting question. They run up to the car, I think they do, but summer before last, we had a small group, and they were only allowed up to that point to drive to the Fruit Loop tree. It's called Fruit Loop trees, like a drive-in. But we introduced the idea that they could walk to the Fruit Loop tree, and if you walk to it, you could pick a lot more Fruit Loops than if you drive through, it just kind of comes in your window, right? So you only get one. And so I wanted to put them to the test. So you can probably imagine where this is going. We had the rat car on one end of the arena, the Fruit Loop tree here, and we put the rat in the middle. So the most efficient path was to walk to the Fruit Loop tree and pick as many Fruit Loops as you can. But two of the three did this. Let's see, I think I can. Went out of its way, not walked, but ran to that car and drove to the Fruit Loop tree. And we repeated this for about 4 weeks, and those animals continued. So there are individual differences. One wanted the shortest path and the more Fruit Loops, but it suggested, and in my TEDx talk, I said, I taught rats to drive cars, and they're teaching me to enjoy the ride, and that really is true. It's humbling when a rat tells you how important hope and excitement is, but that has happened. At my late stage of my career, I feel like I'm just getting started, but it's more important to try to translate some of this information. We're still continuing with our rats, but starting this initiative, Brain Translational Research, Education, and Experience, the Brain Tree Initiative. Our first passion project is, we want to apply what we know about neuroplasticity and environments to individuals who are incarcerated. Is that information being translated so that we truly can restore, reform brains? So we're passionate about that, and I'm passionate about my students. My research would not be done without them. Passionate about the animals and the support from the University of Richmond. Thank you for your attention. If you're not asleep, I'd love questions if you have any. Yeah.
When it comes to the difference in stress levels and neuroplasticity between the wild rats and the lab rats, have you taken into consideration all the natural predators that the wild rat has to worry about constantly the whole time it's out, as opposed to the lab rat that never has to worry about, and the different foods it has to be able to find, while the lab rats can only one kind?
So you're seeing why no one works with these wild rats, all these questions. So you bring up a really good point that an enriched environment in the lab is not a natural environment, because if it was a natural environment, we'd have to have predators. So the predators, it's important. What I didn't say, we did do a study. They let me keep the rats for 5 days out in the hut in the woods. And so the students were able to collect the fecal samples. Now, I thought having a wild animal in captivity, their stress hormones would remain high. After 1 day, they plummeted, they went down. I think they can read the room really well. They had wild animals like squirrels trying to get in, and they couldn't. I had a really cushy nest box and some good food, and I could see them saying, I'm anthropomorphizing, but this is a great Airbnb. For once in my life, I'm not preyed upon, and their stress hormones went down. So it's important to know that, yes, they have the predators, so that's why they have that, but how do they handle it? Are they able to manage it and read the room? Diet. So when we control the lab animals, we feed them the same, like the nuts that you would have for squirrels and such, but that's just the time we have them. We're not able to control that diet prior to that. And it was interesting, we write grants and they get rejected, but I'm trying to get funding for the wild rats, and one of the reviewers said, this isn't good science because the wild rats are stressed all the time and humans are not. That's a very first world.
It's a different type.
Yeah.
They have a primal stress, whereas our stress is developed. We've now incorporated everything negative into the same aspect that we notice negative first, based off of those primal instincts.
Yeah, it's interesting. I think what you say makes a good point, but we carry around somewhat of the brain of our ancestors too. So trying to pick apart what is adaptive stress, maladaptive, I think there's a lot more that we need to know about that, but good question. Other questions about rats? Raccoons, yeah. I'll go over here first. Yeah. Uh-huh.
I just wondered if you've seen any of the stuff about the rats that were taught to play hide and seek right, because they didn't get any food or anything other than just like the joy of playing.
Good for you for knowing that study. And Michael Brett in Germany, he taught rats to play hide and seek with humans. It's kind of complicated, but they kind of knew when they were supposed to hide and knew when they were when they found something. And so I did interview him for Wild Brain's book, and it's hard, but they have evidence to suggest that they were very motivated to do it, and it seemed to be a healthy.
And they made happy noises.
Yeah, those chirps, I know, say, Yak Pinks have talked about it being laughter, but it's the same chirp they make during sexual behavior and eating, so some of the happy, good behaviors. But I love, that was in science. It's such a great, I have a picture of peeking around the creative science, so we kind of bond that way, rats driving and rats playing hide and seek, doing some things beyond the Skinner box, I guess. So we had, yeah. Mm-hmm.
When you were talking about monitoring cortisol levels and said that you were doing that by collecting poop samples and things like that and studying it that way, it made me think about something that's been really big for the past decade or so, and that's the Human Microbiome Project, or just microbiome studies in general. And one of the things that I remember reading about there is that there might be some kind of modulation between the gut and the brain in terms of regulating different, important neurochemicals and so forth. Were you able to study any of that, the microbiota influence on any of that? Did that come up at all in your
Great
work?
question. That's a hot topic. We want to look at, obviously there are going to be huge microbiome differences between the wild and the lab, so we're starting that work. But I didn't tell you about a study. This blew my mind. So I heard the researcher, she was Italian neuroscientist, and when I was at the International Behavioral Neuroscience Society, she reported these findings that, so she took the poop from enriched rats, and there's this thing called a fecal transplant. I don't know if you want to sign up for that, but implanted it in the standard lab animals, and they got the same neuroplasticity. So I don't think it's all about, engagement's still good. Let's just not take, probiotics are good too. But it tells us that it's doing something, some signaling about variability or something that we need to explore further. So there's a lot more of this enriched environment that we need to, but good question. Other hands that were up? Yes, in the back, and then we'll come to you, yeah.
Have you ever thought about doing research on Because I really like cats.
I do too. Yeah.
Yeah, but I wonder if the rats would have any sort of reaction to word association.
Yeah, I mean, that's an interesting question. They do make vocalizations that we can hear. Like the wild rats, they'll scream at me like nobody's business. I don't need a microphone for that, to hear that. But this intricate, complex ultrasonic communication, we just don't know. We have these two very broad categories: fear, defense, and then joy, happy, laughter, whatever that is. But in between, and the duration and the altitude of those, there's probably a hidden language in there. But they also use, olfaction, then pheromones, and different behavioral. They have some facial, like they put their ears back and their whiskers go back. So there's probably a lot of different ways that they can communicate. But it is kind of limiting with a rat. I know there's more interesting research with language with some of them.
They also will vibrate their eyes when they're excited. I don't know if you ever saw that in your study.
I haven't. When a female is flirting with a male, she'll wiggle her ears in an interesting way, like a little helicopter. But I haven't seen the eyes. I'll have to look at that. Thanks for that. And then you had a question. Yeah.
Yeah, I'm just interested in how did you figure out the dosage of hope? Like, how much hope goes to how many grams of Coke? And also when 15 minutes seems like a long time to sustain hope for a rat. Like, that's kind of amazing to me. I don't know if I'm hopeful.
Can you wait? It's like the marshmallow test or something for rats. A good question. So we kind of dose, and now we have the anticipation group. They wait for it, and then a group that gets it all at once. But they have some. You can't take anticipate if it gets it all at once. It's waiting for that one time. And I should say that our anticipation is against the background of unpredictable. They never know when it's going to happen. So we're really highlighting that. And then just a control group that doesn't get any of that. So we kind of look at doses like that. But we're just beginning, and even thinking about dosing behavioraceuticals. I'll go back to Darwin, and I got to go to Down House a couple of years ago. He talked about having a lot of anxiety because you may have heard of this theory he was writing, right? Natural selection, and it, had some, and his wife was very religious, and he had what sounds like irritable bowel syndrome for a long time. But he said that he had this walk, he called it the thinking path around Down House, around his garden. And high anxiety days, he would walk around it like 3 times, and he would put a little stone to remind him. So he had like a 3 walking path dose, or a 2, or a 1. We're just exploring. I just made up the word behavioraceuticals to make behavior and cognitive, those types of therapies, sound, because they are as important as the pharmaceutical. In our Western world, we just really recognize and value the drugs and the surgeries. So what can we do? Because it does change this cocktail, but we're just at the beginning, and I'm just using dose very lightly in a general way, but I think there's a lot of potential to think about it. Yes?
Have you ever been tempted to look at the various genetic aspects of mice?
Of mice?
All the work that Jackson.
Yeah, the transgenic kind of. I haven't had mice in the lab lately, but we have worked with the National Institute of Mental Health with the driving project, actually, to look at transgenic rats that don't produce new. You can give them a medication where they no longer produce more new cells, so it kind of cuts off their neuroplasticity in that realm. Surprisingly, they were better drivers. Maybe it's a no-brainer driving, but we don't think they were as distracted as the animals that could take in more. So that's one thing that we're looking at, but we're also very interested in not changing genetics, but looking at how genetics are different in the wild and the lab animals. What has changed, this epigenetics, how our environments can change genetic expression. So that's what we're excited about, looking at some of that flexibility, but that's a hot topic, so everyone is interested in that.
The mouse models have as much criticism as you had for the lab.
Yeah, and again, I think we need the laboratory work, and we need the convenience. We just need to add to it. And we're building our CVs with these studies, but I'm more interested in what are we doing that may help improve the experiences of these individuals. Now you can say that more easily after tenure and things like that, but that's because you really can't survive doing these weird things like I do. Anything else? You guys are a great audience, and I don't want to keep you here too long. So, all right. Thank you very much.